All right, let's explore. Number 8. The brain's hidden drain. In October 2013, neuroscientist Maiken Nedergaard at the University of Rochester published a paper in Science that forced neurologists to rewrite their textbooks. She had discovered something inside the human brain that, by every existing principle of anatomy, was not supposed to be there. A previously unknown waste clearance system, running through channels surrounding the brain's blood vessels, silently flushing out toxic debris while you sleep. For decades, the brain was considered a uniquely privileged organ, sealed behind the blood-brain barrier, managing its own affairs without the lymphatic drainage system that services the rest of the body. Researchers knew the brain produced metabolic waste. They simply assumed it handled that waste in ways too subtle to detect. Nobody was looking for a hidden plumbing network, because nobody believed one could exist. Nedergaard named it the glymphatic system, and what it does is deeply unsettling in its implications. During deep sleep, it becomes 10 times more active than during waking hours, pumping cerebrospinal fluid through those channels and clearing out accumulations of amyloid beta and tau proteins, the same proteins that form the plaques and tangles found in Alzheimer's disease. The system doesn't trickle, it flushes. Subsequent research has shown that glymphatic function appears to collapse in early Alzheimer's patients, and that chronic sleep deprivation significantly impairs the system's ability to clear these proteins. Most neurologists practicing today completed their training before this system existed in the literature. It was not in their textbooks. It was not in their curricula. The disturbing possibility now being explored is that we spent half a century treating sleep deprivation as a productivity problem, while the brain's only mechanism for washing away the proteins of Alzheimer's disease was quietly failing. What exactly have we done to a generation's brains by celebrating people who sleep the least? Number 7. The gut and the will to live. In February 2019, a team of researchers led by Jeroen Reis at KU Leuven in Belgium published findings in Nature Microbiology that several journals had hesitated to touch. After analyzing gut bacteria samples from over 1,000 participants in the Flemish Gut Flora project, they identified 2 specific bacterial genera that were consistently and significantly absent in people experiencing severe depression. The finding was precise. The absence was not random. The bacteria in question were Coprococcus and Dialister. Their depletion correlated with depression across the dataset even after controlling for antidepressant use, which itself is known to alter gut composition. The researchers were not simply finding that depressed people have different gut bacteria. They were finding that specific bacterial species appeared to be load-bearing components of mental health in a way that nobody had anticipated. The mechanism is not metaphorical. The gut produces approximately 90% of the body's serotonin. The enteric nervous system lining the digestive tract contains more than 100 million neurons, communicating directly with the brain via the vagus nerve in a two-way pathway researchers now call the gut-brain axis. When scientists raised germ-free mice with no gut bacteria at all, those animals exhibited extreme anxiety behaviors and abnormal stress hormone levels from birth. The finding that disturbs researchers most is what happens when you transfer gut bacteria from depressed humans into germ-free rats. Within weeks, those rats begin displaying measurable depressive behaviors. The bacteria appear to be carrying something across species, something behavioral, something that looks, in animal models, like despair. The idea that the will to live might have a microbial component is not yet a clinical claim that would survive full peer review scrutiny. But the hypothesis now has animal data, correlation data, and a growing mechanistic framework behind it, and almost no clinician in the world is trained to act on any of it. Number 6. The switch medicine forgot to flip. In 2016, neurosurgeon Kevin Tracy at the Feinstein Institute for Medical Research in New York implanted a small electronic device into a Dutch woman named Katinka Simonse. She had severe rheumatoid arthritis that had failed every available drug. 7 days after the device was activated, her inflammation markers had dropped to near normal levels. She had not received a new drug. She had received electrical pulses sent through a single nerve. The nerve was the vagus nerve, running from the brainstem through the chest and abdomen and interfacing with nearly every major organ in the body. Tracy had first published his concept of the inflammatory reflex in Nature in 2002, arguing that this nerve carried a previously unknown signal that told the immune system to stand down. The medical establishment was largely skeptical. The idea that a nerve could function as an on-off switch for systemic inflammation seemed too simple, too elegant, and too inconvenient for a pharmaceutical industry built on cytokine-blocking biologics. The 2016 clinical trial, published in the Proceedings of the National Academy of Sciences, changed that. Patients with rheumatoid arthritis who had not responded to methotrexate or biologic drugs showed significant reductions in inflammation after vagus nerve stimulation. The mechanism is now confirmed. Electrical stimulation of the vagus nerve triggers the spleen to halt macrophage production of TNF-alpha, the primary inflammatory driver in most autoimmune disease. The same pathway appears disrupted in lupus, Crohn's disease, type 2 diabetes, and sepsis. The FDA has granted SetPoint Medical a breakthrough device designation for this approach. The science is no longer fringe. It is published, replicated, and device approved. If the body has a built-in anti-inflammatory system controlled by a single nerve, and that nerve can be stimulated without a prescription through breathing techniques and other accessible interventions being studied in parallel trials, why is this still the subject of research papers rather than the first conversation a physician has with every autoimmune patient? Number 5. The cancer clock. In 2022, a paper published in Science by Ludmil Alexandrov and his team at UC San Diego proposed something that overturned the foundational story cancer biology has operated under for 40 years. Alexandrov's team analyzed over 2,500 cancer genomes from 24 different tumor types, drawn from the ICGC and TCGA pan-cancer analysis project. They found a pattern encoded in those tumors that nobody expected. Many cancers do not accumulate genetic mutations randomly. They accumulate them on a biological clock. The dominant paradigm in cancer prevention has been built on the carcinogen model. Avoid smoking, avoid radiation, avoid processed foods, limit alcohol, reduce your exposure to the environmental agents that damage DNA. This model is real. It is correct for a meaningful proportion of cancer cases. But Alexandrov's team identified specific mutational signatures. They labeled Signature 1 and Signature 5 that appear in almost every cancer type and accumulate at a constant, predictable rate, tied not to carcinogen exposure, but to the simple passage of cellular time. These clock-like signatures increased linearly, with the patient's age regardless of lifestyle, smoking status, or environmental history. The cells were accumulating damage at a rate set by biology itself, not by behavior. The mutations arrived on schedule, the way a clock hand moves, independent of what the organism was doing. The disturbing implication being modeled by researchers is that some percentage of cancer risk is mathematically encoded in the process of cellular aging and cannot be meaningfully reduced below a threshold set by how old the body is. The carcinogen model addresses what the body encounters. The clock model reveals what the body does to itself. The cancer clock doesn't stop because you eat well. It doesn't stop because you quit smoking. It runs in every dividing cell in every living body. And if the current models are correct, some percentage of cancer deaths were never preventable through lifestyle changes. They were only ever detectable early enough to matter. Number 4. The darkness we call anesthesia. Every year, approximately 313 million surgical procedures are performed worldwide under general anesthesia. Every year, anesthesiologists administer drugs that render patients unconscious in under 2 minutes. And every year, not one of those anesthesiologists can explain exactly how those drugs work. This is not a gap in training or a failure of communication. It is a formal admission embedded in the scientific literature, stated explicitly in review articles published in Anesthesiology and Nature Reviews Neuroscience. General anesthesia has been practiced since October 16, 1846, when William Morton demonstrated it at Massachusetts General Hospital. In the 178 years since that demonstration, the drugs have been refined, the delivery systems perfected, and the monitoring made more sophisticated. The mechanism of consciousness suppression has remained unknown. The leading hypothesis involves disruption of thalamocortical communication, the signaling loop between the thalamus and cortex that appears to underlie conscious awareness. But this hypothesis cannot explain why anesthetic agents with completely different molecular structures all produce the same result. What makes this stranger still is the breadth of organisms affected. Canorhabditis elegans, a nematode worm with 302 neurons and no cortex whatsoever, responds to anesthetic agents the same way humans do. The mechanism predates the vertebrate nervous system by hundreds of millions of years, and we still do not know what it is. The clinical consequence of this ignorance has a name. Anesthesia awareness is the phenomenon in which patients are conscious but paralyzed during surgery. Multiple meta-analyses confirm it occurs in approximately 1 to 2 patients per 1,000 procedures. EEG-based monitoring tools designed to detect it have documented failure rates. There is no reliable real-time confirmation that a patient is actually unconscious. The next time someone you love is wheeled into an operating room, consider this. The most fundamental thing about that procedure, the part designed to ensure they feel nothing, is built on a mechanism that, after 178 years of practice, medicine still cannot explain. Number 3. The brain's uninvited tenant. In 2016, Rudolph Tanzi and Robert Moyer at Harvard Medical School published a paper in Science Translational Medicine that reframed the foundational understanding of Alzheimer's disease. Amyloid beta, the protein that forms plaques in Alzheimer's brains, and that pharmaceutical companies had spent 20 years and more than $40 billion trying to eliminate, was not a disease product, they argued. It was a defense mechanism. The brain was not failing to clear these plaques. The brain was building them deliberately to trap and neutralize pathogens. The amyloid beta protein, they showed, has potent antimicrobial properties. It forms sticky nets capable of capturing and killing bacteria, fungi, and viruses, including herpes simplex virus type 1. A follow-up paper published in Neuron in 2018 demonstrated that mice infected with herpes simplex virus rapidly developed amyloid plaques, and that those plaques suppressed viral spread throughout the brain. The amyloid was functioning as intended. It was the immune system doing exactly what immune systems do. The epidemiological data has steadily strengthened this framework. Taiwanese cohort studies published in journals, including JAMA, found that patients who received antiviral treatment for herpes simplex infections showed risk reductions for Alzheimer's of up to 90% in some analyses. Postmortem examination of Alzheimer's brain tissue has detected Porphyromonas gingivalis, the primary bacterium in gum disease, as well as Candida species and cytomegalovirus across multiple independent studies. More than 200 clinical trials have targeted amyloid beta for removal in Alzheimer's patients. More than 200 have failed. The failure rate exceeds 99%. Researchers at Cortexime conducted phase 2 trials of an antibiotic targeting Porphyromonas gingivalis in Alzheimer's patients before funding collapsed in 2021, with results that were inconclusive rather than negative. 200 failed clinical trials targeting the same protein should have triggered a fundamental reassessment of the hypothesis. Instead, the field largely persisted. The researchers arguing that Alzheimer's begins with a pathogen are not working at the fringe. They are at Harvard, and for nearly a decade, the field has found ways to look past them. Number 2. The trade we never agreed to. In November 1989, epidemiologist David Strachan published a paper in the British Medical Journal with a hypothesis so counterintuitive it was initially treated as a curiosity. He had noticed that children with more older siblings had significantly lower rates of hay fever and allergies. His proposed explanation was precise and unsettling. The immune system requires exposure to infection during early development to calibrate correctly. A childhood without sufficient microbial contact does not produce a healthy immune system. It produces a dysregulated one, armed and trained, with nothing appropriate to fight. Strachan's hygiene hypothesis was later refined by Graham Rooke at University College London into what he called the Old Friends hypothesis, published in Nature Reviews Immunology in 2003. The distinction matters. It is not simply cleanliness that disrupts immune development. It is the absence of specific ancient organisms, bacteria, parasites, and environmental microbes that co-evolved with the human immune system over hundreds of thousands of years, and whose presence the immune system requires to learn the difference between a genuine threat and a harmless stimulus. The epidemiological signal is enormous. Autoimmune diseases including type 1 diabetes, multiple sclerosis, rheumatoid arthritis, and inflammatory bowel disease have doubled or tripled in developed nations since the 1950s, while remaining stable or lower in nations with higher pathogen exposure. Rates of childhood peanut allergy in the United States increased 21% between 2010 and 2017 alone. In 2015, a landmark study published in the New England Journal of Medicine found that Israeli children exposed to peanut products as infants had an 81% lower rate of peanut allergy than British children in whom peanut avoidance had been standard medical advice for years. The American Academy of Pediatrics reversed that guidance in 2017. The official recommendation had been accelerating the very disease it sought to prevent. Caesarean-born children carry measurably different gut microbiomes than vaginally born children and show higher rates of asthma, allergy, and type 1 diabetes in multiple independent cohort studies, attributed to missing bacterial colonization that would have occurred during birth. The theory that immunologists are reluctant to state plainly is this: we did not simply eliminate infectious disease in the developed world. We traded it for inflammatory disease, and we are only beginning to calculate the scale of that exchange. The researchers building this case are not proposing that infections were good. They are demonstrating, with decades of converging data, that the microbes we eliminated were doing something essential that nothing in modern medicine has replaced. Number 1. The infection beneath everything. In 1983, Australian physician Barry Marshall did something that violated every standard of medical ethics and basic common sense. He drank a beaker of Helicobacter pylori bacteria. Within days, he developed gastritis. Within 2 weeks, he had the beginnings of a peptic ulcer. He then cured himself with antibiotics, documented the entire process, and submitted his findings to a medical establishment that had already decided stomach ulcers were caused by stress and excess stomach acid. He had been told that for years. He had seen the data proving otherwise. Nobody would listen. Marshall and his colleague Robin Warren had been arguing since the early 1980s that the most common gastrointestinal disease in the developed world was not a disease of personality or lifestyle, but a bacterial infection, treatable with a course of antibiotics costing a few dollars. The pharmaceutical industry had built an enormous infrastructure around acid-suppressing drugs. The gastroenterology field had a settled consensus. Marshall drank the bacteria because no other path to being believed was available to him. He and Warren received the Nobel Prize in Physiology or Medicine in 2005. H. pylori is now classified as a Group 1 carcinogen by the International Agency for Research on Cancer. The settled consensus had been wrong for decades, and the cost of that wrongness was measured in suffering and in deaths. The question that a growing number of researchers in virology, oncology, neurology, and cardiology are now asking carefully in peer-reviewed literature, one paper at a time, is whether the Marshall precedent is not an isolated historical curiosity, but a preview of something much larger. What if the chronic diseases currently understood as metabolic, degenerative, or genetic conditions have infectious origins that were missed because they did not present like infections? Not acute, not febrile, not contagious in the obvious sense, but slow and latent and inflammatory, triggered by pathogens that medicine spent decades looking for in the wrong places. The evidence being assembled is specific. Fusobacterium nucleatum, an oral bacterium, is consistently detected in colorectal cancer tissue and associated with worse prognosis across multiple studies from Wendy Garrett's lab at Harvard, published in Cell Host and Microbe. Cytomegalovirus seropositivity is associated with increased arterial stiffness and atherosclerosis in multiple independent cohort studies. Chlamydia pneumonia has been detected in arterial plaque tissue since the 1990s, enough to prompt large-scale antibiotic trials in cardiovascular disease. Then, in January 2022, a study led by Harvard epidemiologist Alberto Ascario and published in Science followed 10 million United States military personnel and found that Epstein-Barr virus infection preceded the onset of multiple sclerosis in essentially every case examined, with a 32-fold increase in MS risk following EBV infection. Researchers commenting on the paper described it as the closest thing to proof of causation yet demonstrated for any autoimmune disease. Multiple sclerosis, for which there is no cure, may begin with a virus that infects nearly everyone and causes a catastrophic immune misdirection in a subset of those infected. The infectious origins of chronic disease hypothesis does not argue that lifestyle is irrelevant, or that genetics do not matter, or that every cancer and every heart attack and every case of Alzheimer's begins with a pathogen. It argues something more precise and more disturbing. It argues that a significant proportion of the deaths and disability attributed to the leading chronic diseases of the developed world may involve infectious triggers that were overlooked because medicine built its entire treatment infrastructure around a different assumption, and assumptions of that size do not fall without taking institutions with them. Barry Marshall drank the bacteria in 1983. He won the Nobel Prize in 2005. The gap between those two dates was not a scientific gap. It was an institutional one. The researchers now building the case that heart disease, cancer, multiple sclerosis, and Alzheimer's have infectious components are not publishing at the fringe. They are publishing in Science and Cell and Nature and the New England Journal of Medicine. The gap between their findings and what happens in a doctor's office today may turn out to be the most consequential institutional gap in the history of medicine. And unlike the ulcer story, this time there is no single beaker anyone can drink to force the world to pay attention. If you want to see more videos like this, click the video on screen now and make sure to subscribe.